Mendelian Genetics. In the previous video, we looked at the results of meiosis. Okay, in the video before that, we looked at the results of mitosis, right? So just a very, very quick review. The results of meiosis was that there were 4 haploid gametes, remember? So a haploid meant that the chromosome number will be represented as just N without a coefficient of 2, okay? And the gametes are the sex cells, right? And there are different combinations of the alleles for the different genes. Along the chromosomes. And remember, alleles make up the genes, right? So if a gene was pertaining to like eye color, an allele would be brown eyes, okay, or blue eyes. And obviously you can only have one of these alleles for that particular gene characteristic, right? In this case, we're talking about eye color. So obviously we only have one allele of each of these genes, right? So there's one allele of each gene. But obviously these are haploid, right? So when a sex cell, let's say a sperm cell meets an egg cell, the other gamete cell, fertilization occurs, and then of course it reestablishes that diploid, right? That 2N, right? So fertilization reestablishes the diploid number, okay, the number of chromosomes, because it produces that zygote, right? That first step before becoming that fetus, and then it's going to grow into a larger being. All right, thanks to those 2 sets of chromosomes, right, from the egg and sperm combined. During fertilization, it is random, right? So I'll just put here random. So the chromosomes from the mom and the chromosomes from the dad are then assorted in a variety of ways. Okay, so let's do a little bit of math here. And remember we had 23 pairs, right? So if we have 23 pairs of chromosomes, obviously a pair is represented by the number 2, right? So then we obviously have to put that as the base of the exponent, right? So if we have 2, okay, represented as the pair, and how many possible combinations are in an egg or a sperm, right? So in a gamete, right? How many possible combinations are there? There's 23, right? Because it can make 23 other pairs. That gives you a number of 8,388,608 combinations, right? Again, this is for the sperm or the egg, okay? Whichever one you're talking about. So let's say this one is for the sperm, okay? What is the possible number of combinations for an egg then? Well, it would be the same thing. So again, a pair to the power of the possible combinations. So it would be a 23, and it would be the same number, right? So we obviously have to do something with these two, right? The sperm number and then the egg number of possible combinations. And obviously we have to cross them, right? You wouldn't add them, right? We're literally trying to cross the amount of combinations that you have in one gamete with other possible combinations in particular orders with the other gametes, right? So it wouldn't make sense to add. You would obviously have to multiply. So you end up getting a very, very large number. It would be 70,368,744,177,664 possible combinations in an offspring. All right, so this is for the offspring. All right, if you want to be even more precise, we can say the zygote, right? All right, so therefore it generates a new combination. Of genes when they're located on different chromosomes. So what is this all pretty much just trying to tell us? With so much variation, it's pretty much impossible for your parents to create another you. You are very, very unique. So just to conclude with meiosis segment, was that individuals, so this is individuals, are produced by the fusion of two gametes, all right, the sex cells. So they are genetically variable because they get this genetic info from both parents, right? All right, so what are some disadvantages? Okay, so what are some cons here? There needs to have specialized organs to produce these gametes, all right, these sex cells. And sexual selection practices can drive attention. A very common example are like peacocks, right? Especially the male peacocks. They're very, very bright and diverse in color and have very, very large feathers in order to drive attention for the female, right? But this can also make it very difficult to run away from predators and can even be easier to spot, right? So it's very risky. But obviously a pro is that there's an increase in variance, which also increases resilience to particular diseases. Now let's quickly touch upon mitosis bring so that we can bring those two concepts together and get into Mendelian genetics. So this one produces an individual from just one parent. All right, so no sex cells are involved here. Okay, and the chromosomes of the parent cell are duplicated and divides. So each daughter cell receives that one copy of each chromosome. Okay, and obviously each daughter cell is genetically identical. Genetically identical. And a pro for this is obviously no need to seek a mate. So it's very energy efficient, okay? It's not costly or taxing. But obviously an issue is that it's invariable, so it is vulnerable to diseases easier. Now let's bring those concepts that we just quickly reviewed from mitosis and meiosis to this karyotype, all right? So what you see here is a karyotype, and it's a snapshot during a stage in the cell cycle, and it's able to display all the chromosomes in a cell. So in mitosis that we just looked at, they're responsible for the body cells, all right? Body cells undergo mitosis. So literally everything here. And as you can see, there are 22 pairs, all right? 1 all the way to 22. And obviously a pair has 2 chromosomes each. So 22 times 2 gives you 44 chromosomes. And again, these are only for the body cells, all right? Or autosomal. However, you should know that in a diploid, we have 46 chromosomes, all right? So where are the other 2? They're going to be right here. This will be considered as the 23rd pair, okay? So in a female, there is an X and X, so we can put one there, one over there. But if it's a male, there's a chromosome belongs on the X and another chromosome on the Y. All right, so the Y chromosome is actually a lot smaller than the X chromosome, so just keep that in mind. So obviously there's only 2 chromosomes pertaining to that 23rd pair, okay? Whereas there's 44 for the autosomes. And I just want you to remember that the homologous chromosomes that are the same size and shape and consist of similar loci of genes are paired together, okay? With the largest pair being at chromosome 1 and the smallest pair at chromosome 22. This is an individual that is healthy because there are 46 chromosomes in total, and all of these chromosome pairs are of similar size and shape, okay? Also, there's no +1 or -1 or plus a few or minus few chromosomes on any of these pairs. So as long as those are met, it is indicative that it is healthy. So just note that the chromosomes don't have to be perfectly symmetrical or identical in appearance as its pair for it to be considered a healthy pair. Okay, sometimes they might look a little bit off, a little bit different, but as long as they're roughly the same size and shape, they're considered healthy. So snapshots in time for karyotypes aren't always perfect because obviously it is undergoing division during this time. And obviously there's organelles and particles around within the cell, so it can get sometimes messy when it comes to the photographs. So yeah, if they're roughly the same appearance, you would consider them healthy. If there was an extra one over here, or let's say the Y chromosome here got removed, then those are examples of it not being healthy. All right, so let's get into the Mendelian genetics. Obviously, genetics is the study of inheritance, of passing down genetic information of traits from parent to offspring, and then the generation after that, and the generation after that, etc. So some key vocabulary for you to remember. So for a gene, remember it's that section of DNA, right? Section of DNA associated with a trait. Okay, but then what is a trait then? It's like a characteristic that is inherited. All right, from the parent. All right, an example was like, let's say eye color again. So what makes up those eye colors, right, that we already looked at, brown, blue, hazel eye colors, those would be alleles because they are a form of that gene. All right, blue eyes, brown eyes, etc. Now let's look at a phenotype and a genotype. What's the difference? A phenotype is literally a physical appearance of a trait. All right, so what's the trait, for example? So let's say eye color. So let's say that person has the allele for blue eyes. So can you see that a person in front of you has blue eyes? Obviously, right? So then you can literally see that the person physically embodies that trait. Anyone can see it. So that's what phenotype represents. It's what you literally can see. A genotype, however, is different. You can't see it. This would be a description of the alleles that the person carries, okay, for that same trait. So if we were to tag along on just eye color, okay, because we did that for phenotype, so let's just do the same thing for genotype. How are they coded for blue eyes? Obviously, you can see that they have blue eyes, but how is it coded that they have blue eyes? So for example, let's say blue is represented as a small b, all right? But a brown eye could be represented as a big B. If they have blue eyes, they need to have two of these small b's. Because if the individual has only one small b but has a big B, guess what? This person doesn't have blue eyes. This person has brown eyes because big B is a capital, right? Small b is a lowercase. So capital always outweighs or is more dominant than the lowercase. But if this person has a small b and small b, because they have blue eyes, you can literally physically tell phenotypically that they have blue eyes. How about their parents? Do both of their parents have blue eyes? So we could do a little crossover here. Let's say the dad is up here. Let's say their dad had a big B, small b. And let's say that we have the mom over here, okay? Let's say she also had a big B, small b. So both the parents had brown eyes, okay? But if you cross them, you get big B, big B. So this is obviously brown-eyed, a chance of having a brown-eyed offspring. We got big B, small b. Again, another chance of having a brown-eyed offspring because the big B is a capital. It's stronger and more dominant than the small b, which is the blue one. And then over here, big B, small b. Same thing, brown eye, offspring chance. And then the last one here, small b, intersecting, which would be a blue eye. So that would explain how that person had blue eyes phenotypically. But genotypically, we represent their code of alleles as small b, because if blue is represented as a small b, because blue is a recessive eye color trait, okay, it's weaker than the brown eye, we can genotypically tell that this code that they received was hidden because it was carried by the dad's second allele and the mom's second allele. So all these things that we're doing over here based on code and what we're crossing over here is an example of a genotype, of a genotypic test, because a genotype is a description of the alleles. So again, phenotype is that they have blue eyes, clear as day that you can see. A genotype is small b, small b. That is that description of those alleles for that specific trait of blue eyes. So I know I just used this word dominant. So it's pretty much when an allele overpowers another. All right, so obviously an example of that, okay, represented as capital letter. A recessive is the weaker one. All right, this is when the allele is masked. You can't see it when a dominant is present. So that blue-eyed person, you can see that they have blue eyes that you can literally see, right? But what you cannot see is a small b alleles. That's what you cannot see. And where did this thing come from? It came from the dad's big B, small b, and the mom's big B, small b. So which one is recessive here? Which allele is recessive? Obviously this one and this one is recessive. Because the parents had brown eyes. Because this big B and this big B here were dominant. All right, so this one were dominant. But again, that small b and that small b here was able to be passed on despite being hidden the whole time from their parents. Now, when something is homozygous, this means that the genotype is where both alleles are identical. So exactly what we just did. Small b. This represents blue eyes, right? Homo means the same, right? So this is recessive, it's a small b. This is recessive, it's a small b. So that's why this is homozygous. Heterozygous is the opposite. It's the genotype where the two alleles are different. Remember the example of the brown-eyed parents, both the parents, okay? Obviously, this one over here is dominant, capital letter. This one here is lowercase, so recessive, so weaker. Because they're different, this is heterozygous. Hetero means different, right? And again, remember this was brown-eyed. So in heterozygous pairings, the dominant one is the only one that is showed, okay? So remember, big B represented brown, small b represented blue. Because B is stronger, you can only see the brown eye trait. So let's look at Gregor Mendel, okay, and his first experiment. So he was the first to experiment with inheritance patterns, okay, a very, very long time ago. He planted these pea plants that you can see over here, right? This is a very tall pea plant, this is a very small pea plant. And he observed the growth, okay? And normally pea plants, they self-fertilize. However, he just manipulated them and changed it to allow for cross-pollination. So obviously, you know what we're going to be doing. We're going to cross these, okay? So if we eventually ended up getting 4 offsprings, okay? what do you think they became? Do you think they became tall, or do you think they became short, or do you think they kind of were in the middle, like medium-sized? It turns out all four offspring ended up being a tall plant. Okay, every single one of them. So I want you to pause the video and think about why. Okay, based on the excerpt that I've done in the earlier on in this video with the blue eyes and the brown eyes, how do you think every single one turned out to be tall? Use that cross that I briefly talked about. Okay, give it a try on your own and then come back and then click play again. So let's say this one up here was one of the parents. So this is the P generation, by the way. Okay, the P generation. So let's say the tall one was big T, big T. All right, and then the small plant here was small t, small t. You end up getting big T, small t, big T, small t, big T, small t, big T, small t. So it makes sense if we were to do it this way, that all of these would have ended up as a big T, small t. So tall, as you can see over here, makes sense. But I hope you drew a second cross possibility, because guess what? What if it was a big T, small t? And obviously this one has to be small t, because that's the only way that the recessive allele can be shown. What if you have big T, small t, small t, small t, big T, small t, small t, small t? That's fine. You can still get 4. Okay? You're probably thinking, wait, there's only 2 over here. So shouldn't only 2 of these be tall, and maybe these ones would be like really small or something? No. Because I want you to think of every single one of these boxes as a particular chance of being that pair of alleles. So out of these 4 possibilities, let's say it lands on this one, okay? 1 out of these 4 chances. That means this plant, let's say this is the first one that got chosen. So let's say this is the first one and it exhibited a big T, small t. That's fine, so that's big T, small t, okay? So that's its genotype. However, this gets reset as opposed to being crossed out. Okay, so it's not like when you do this again that you only have 3 of these possibilities. That's not true. You still have 4 possibilities, but now maybe the small t could be chosen. Okay, but in this case we have no small t's, right? We don't see them anywhere. That means when this got reset after this first round, what if this one got chosen again? Then maybe this one is a big T, small t again, all right? And the whole thing got reset again, okay? And then we have a third plant, all right? But this time maybe that one was selected. So then this would be a big T, small t again. And then this last one ended up being a big T, small t. Okay, so obviously these in this lottery draw were never picked out. So let's do that fourth one here. Maybe this big T, small t box got chosen again, so that's why you see all 4 of these ended up being big T, small t's, because they're all tall phenotypically. So in this example, he was absolutely flabbergasted. Okay, so let's look at his second experiment. All right, so second experiment. These were the offspring pea plants from the previous example. Okay, so this is not the P generation. Remember, P stands for the parent generation. This is now the F1 generation, okay, known as the offspring generation. So the first offspring, okay? So out of those 4 really big tall plants, we've taken 2 of those. All right, so maybe we took this one and this one. All right, and we crossed it. What do you think is going to happen in the F2 generation? So this would be the next generation after the first offspring generation that we tackled. You saw this. Can you pause the video and give this a try for yourself? Okay, use those crosses, okay, to figure out what happened. How come 3 out of the 4 offsprings that were produced were tall, but the one of them became short? And press play when you're ready to see the answer. So hopefully you guessed that this one must have been a big T, small t, and this one must have also been a big T, small t. Why? Because if you did a cross, it would be the only way that you could have this one, a small t, a short pea plant. Okay? Right over here, right? Small t, where they intersect. The other possibilities gives you big T, small t, big T, small t, and big T, small t, all tall plants. Okay, which is fine for these ones, but there is obviously a short plant. So there needs to have a possibility of getting a small t. This is only possible if you do a big T, small t cross with a big T, small t from its previous parent. So obviously these ones were offsprings before, but they are now the parents of this new offspring that we're trying to tackle in this question. So this made him realize the concept of dominants and recessives and how significant of a role that they played in genetics. All right, so let's look at the conclusions of Mendel. All right, so there were three. The first one, quite obvious, that there is a principle of dominance. What I just reiterated, certain traits are dominant over others. All right, so dominant, obviously greater than recessive alleles. The second one is the law of segregation, which states each gamete has an equal probability of getting a particular allele. So again, if we looked at brown eyes and blue eyes again, and it's clear that an individual has two alleles for each trait. So this allele and this allele for this parent, coding for the brown eye over here, and this allele and this allele for this parent, coding for the blue eye. That separates during meiosis. So if we look at every single one of these gametes, big B, small b, small b, small b, big B, small b, small b, small b, this has an equal probability of being chosen in the lottery as everyone else, okay? Because the chances of this being chosen is 25%. But what is the chance of this one being chosen? Also 25%. Same thing with this one, also 25%. But both of these code for the same trait. So the chances of you getting a brown-eyed phenotypic characteristic is actually 50% because you can just add these up because they are common denominators, right? They're exactly the same. They're identical. Same thing could be said for this one, even though this particular one has a 25% chance because they are identical here, right? The small b, small b, small b, small b. You could also add these up and say that there is a 50% chance of getting a blue eye. Okay, so there is no resetting. This is what I'm talking about before, right? There is no resetting. Every time an offspring is chosen, it's not as if it's crossed out, but instead a lottery of 4 chances of probability are handed on a platter again. All right, the last one is the law of independent assortment. And this just means that genes located on different chromosomes assort independently of one another. And the best way to exemplify this, in my opinion, is by using a question. Okay, and this question is going a little bit advanced, but it really helps understanding this concept. So when it says located on different chromosomes, okay, the genes, because the color of the eyes are very different than the type of earlobe. So this is obviously that they are on different loci of the genes, okay, on the actual chromosome. So it says Paul has heterozygous brown eyes, all right, so I'll put Paul up here. Heterozygous, okay, that means it's going to be different letters for the alleles. So it means the alleles are not going to be the same. So when we look at brown eyes, remember that's dominant over the blue eyes, okay, compared to the wife. So obviously Paul has big B, small b. Again, I'll just put it as a reminder up here that big B is for brown eyes and a small b is for the blue eyes. Okay, but what else? He has heterozygous detached earlobes, right? And I want you to know that detached earlobes are more common, they're more dominant. So we'll put detached, okay? Whereas the attached earlobes are a small e, so they're recessive. But in any case, Paul is heterozygous here as well, so obviously we'll have a big capital letter and a small lowercase letter. So we can put big E, small e. So if we look at the wife now, wife is homozygous. Okay, homozygous means they're the same letter. Okay, so it means that they're the same allele type, right? So homozygous what? Is it big B, or is it small b, small b? Okay, it says recessive blue eyes. Okay, so obviously we already know blue is recessive. Okay, but it says it here anyway, so we'll just put small b, small b. Okay, it's homozygous, so they're the same, small b, small b. And then heterozygous, all right? So again, heterozygous means capital and a lowercase anyway, okay? So it says detached earlobe, that's pretty obvious. All right, so how are we going to cross them? Well, you can't actually cross them though the way that we've been doing it so far, because this is where that rule comes in, all right, for the law of independent assortment. You actually have to cross these together first, and the wife's different traits, cross them together first, and then you can finally cross Paul and wife together. Let me show you. So you have a big B and a small b crossed with a big E and a small e. Big E and a small e. All right, so you get. So I want to put B's first before the E's, but it's up to you. You can put an E first as well, but I want to put a B first. So big B, big E, and then you have a small b, big E. Then you have a big E, big B, small e, and a small b, small e. Okay? How about the wife though? For the wife, we have small b with a big E, small e. So you get small b, big E, small b, big E, small b, small e, small b, small e. So these are all the genotypes for Paul. All right, you got big B, big E, small b, big E, big B, small e, small b, small e. But for the wife, you can actually group them together because small b, big E is the same as this one. So we'll just put small b, big E over here, okay? We can kind of get rid of that one, okay? Because it's already identical. And then this one too is the same as that, so we can just get rid of this one maybe and then just focus on that one, okay? So you get another small b, small e. So let's attach it here on the side. Get it? Attach it here on the side. Attach. Sorry, that's a little dad joke there. All right, so I've written it here on the side. Let's do our cross. We'll do our final cross, okay? And I need to make it like this. They're very long because I'm going to put Paul's here on the top and then the wife's here on the side. Okay, so the wife's doesn't need that much space. Okay, just needs this space and this space for the two pairs of alleles. So again, Paul has big B, big E, small b, big E, big B, small e, and small b, small e. All right, where the wife's is small b, big E, and we have a small b, small e. All right, so this is where the law of independent assortment comes in. All right, we are now going to cross these together, but the b's go together and the e's go together. Okay, so they still independently assort despite how messy it was in the beginning. Okay, they still get together. That's what it meant. All right, so you got big B, small b with a big E, big E. Then you get a small b, then you get a small b with a big E, big E. Big B, big E, small e, small b, big E, small e. Over here, big B, big E, small b, big E, small e, big B, small e, and small b, small e. So what does this mean? Let's look. What is this? Big B, small b. That is obviously a brown eye. Okay, so there's obviously a brown eye. And what? What's big E, big E? Obviously, big E here is a detached earlobe. Okay, so it's brown eye and detached. All right, so that's this one over here. All right, so we can cross this one out for now. We're done with that. So I'll just put one tally over there. Then how about this one? Small b with a big E, big E. So that is a blue eye. Okay, so we have a blue eye. And what? Okay, big E, big E. Same thing. That's a detached earlobe. Okay, but we can't group these together because obviously this one has a brown eye, this one has a blue eye, so they're not the same. Okay, so we have one there. We're going to put a tally there as well. And we're going to cross that one out. All right, let's look at this one. All right, this one is a brown eye. Okay, with this is a detached. Okay, that's this one over here. So I'll put another tally over there. All right, let's look at this one over here. This is a blue eye, right? So blue eye with a detached. That's exactly what we see over here. So I'll put another tally over there. So get rid of that one. How about this one over here? This is a brown eye with a detached. Okay, that's what we have over here. So I'll put another one over there. All right, this one over here is a blue eye with a detached. Okay, that's this one over here. So I'll put another tally over there. What about this one? This one is a brown eye with an attached. Okay, so I'll put here brown eye and attached. That's one tally. And we're on the last one. This one is a blue eye with a attached. So blue eye and attached. Put another tally over there. So there is a 3 out of 8 chance that their child has brown eyes and detached earlobes. There is a also 3 out of 8 chance that their child will have blue eyes and have detached earlobes. There is a 1 out of 8 chance that they'll have a brown eye, that they'll have brown eyes and attached earlobes, and also 1 out of 8 chance that they'll have blue eyes and attached earlobes. So we just did a dihybrid crossover here, which is awesome, but again, the point of this third finding that Mendel found was that this will independently assort away from this allele's trait and get together with its eye color allele pairing. All right, so big B will go with the small b. Same thing with this big E. It is going to separate away from that eye color traded allele, but it's going to get together with its other big E alike trait. And that's what you saw in these boxes. They have independently assorted to go back together.